Okay, welcome to 10C. This is the germ theory and prevention. We're going to start talking about the germ theory. It's also called Koch's postulates, and this is to correctly identify a causative agent or an agent that causes a problem for any particular disease. So, first one is to take the pathogen and isolate it from an infected critter. In this case, it was a mouse. So we actually put in a hypodermic needle. Remember, hypo goes in and pull out the fluid we want, and this looks like it's blood. And then we actually take that blood to a petri dish that's got some agar or some sort of food source for the bacteria, virus, whatever we're looking for. We let it incubate for a while, and then we let the pathogen grow in the lab. And then we take that pathogen and we go put it into another hypodermic needle to actually put material into a critter, in this case a rat again. It still kills the rat. We pull the material out and we put it back into a lab and we take a look at it, and if we identify it as the original pathogen, we've done our germ theory correctly, and then we can start worrying about cures. Disease cause and prevention. Causes, we can have pathogens, whether they're virus or bacteria. Genetic disorders, we can have diets, exercise, sleep, and then toxins. And usually it's lack of too many, lack of a good diet, and genetic disorders we talked about in one of those units. And preventions, we can do sanitation, try to wash our hands, keep them clean, and aseptic practices. Although I don't want you to start using alcohol-based liquids that say they kill 99.9%, because remember that causes problems. Handling food and water properly, make sure we wash our hands, keep the area clean, try to use chlorinated water that's actually been cleaned of biologic material. Lifestyle choices, always try to do smart things, and then we can take vaccines to actually stop the spread of bacteria or some viruses like polio, which is almost completely eradicated off the planet. Characteristics of viruses, remember this is the dog racer. So D, all viruses have DNA. The problem is now is it just codes for, this is a bacteriophage, which is going to be a virus that affects bacteria. It codes for the protein coat and the rest of the virus, or an influenza virus, which is round. And I hate that when that happens. That was a video I'm making for oceanography. So we also have the same thing here. We got a protein coat and also codes for the DNA that's located inside viral DNA. All viruses are organized, but it's the protein coat and the DNA that's the organization. And growing, viruses do not grow. They take over and reproduce their DNA in other critters. So here's a bacteriophage. These spaceship-looking viruses come in. There's the round prokaryotic DNA free floating in the cytoplasm. There are ribosomes and other things in there, but no real self-contained organelles. The virus of the DNA gets pushed in. It actually deactivates the bacteria of the DNA and actually copies itself, makes new baby viruses that erupt out of the cell membrane and go out to affect more bacteria. So its growth is constructed by the host, not the virus itself. Viruses do not respond to stimulus or maintain homeostasis. Viruses can adapt, but only through mutation, and they do that to survive. And they are not made of cells, which is kind of messes up that cell theory, which is maybe why some people are actually saying that viruses are not alive. Energy, they do not require energy. They rely on others for that reproduction, and they must be replicated in the host cell. Here's a video which I will actually put in. If I click this and we start watching it, I'm going to get another one of those warnings on my video, but I'll put the URL in the comment section of this. And then characteristics of virus. It's got that head, it's got a tail, it's got really funky looking feet, and then it also is able to have this tail go into, this is a bacteriophage, and you can see here are three of them sitting on top of a bacteria with the DNA being pushed in. And here's an electron microscope of the same thing, lots of bacteriophages with a bacterial cell. You don't really see the DNA being pushed in. And here's the influenza, the rounder type DNA that they've actually cut to show the DNA that's actually, or the protein coat, rounded protein coat, that they've cut it to show the DNA. It goes in, does the same thing, goes into the nucleus, deactivates the regular DNA, actually starts getting replications, and then we go out, and remember we have translation, transcription. So transcription inside, translation outside, we start making these new proteins, and the proteins bud, and they start making new mature viruses. And I think that's the end of Unit 10. Thanks for stopping by.